Welcome back, everyone. So I'm going to do a recap of the Extremely Open Science stream for this week. And so, as usual, let's start with a recap of the recap. So let me just share my screen there. And right, so the previous session we had started the process of running the GMGC, so the Global Microbial Gene Catalog, and we had done this splitting of the file into multiple chunks, and I showed this from the JUG paper, where this is a very standard JUG approach where we fan in, fan out. So we break up the task into multiple subtasks, then each one of these can be run even independently. In our case, we're not running anything independently, but it does allow the ability to be much more robust, because as I've said before, I want everything in this project to be potentially runnable by anyone using only a standard laptop. I'm running everything on my laptop, and so we did that, and then I ran it in the meanwhile, and so we had 48 chunks from Run GMGC. And so what we did first during this week is that we wrote this. So this is what we had: split the FASTA, run diamond, and then we just wrote a function that just concatenates everything in the simplest possible way. So this is a very simple function, and this generates this file here. So let's just show this file here. Right, so now we have here a single file with everything from the GMGC. So we got basically the same results as if we had done this big diamond run that we tried the first time, except now this was a little bit more robust, because each one of these 48 tasks took anywhere from 1 to a few hours, and so that meant if something went wrong, my computer had to interrupt it. That was fine. I would just interrupt that one task, and then it would just pick up from there. Right, and then the next thing we did is we now took this, now that we have these results, and actually spent a little bit of time playing with this, but this is a big file that we've been playing around with, and let me just read this. Okay, I'm just gonna start this so we can, and then all right, and so here we load the big GMGC file. So this is the results of this run. So GMGC, so this is just the complete genes actually output, and the same sort of splitting. And actually during the stream we had Antonio actually tell me that this could have been done much simpler using S3.split like this. So this could have been a little bit simpler. I guess this could have been, and then here we are going to be filtering. So we only want to keep KOs such that there's at least the minimum number of unigenes, and so that's here up at the top, 100, and then we filter, and that brings us to, let's just do a little bit since we're here. So this, okay, so that was actually quite a big, so we went, so we only keep a small fraction such that the KO has at least 100 hits. Actually much smaller than I expected, to be honest. Yeah, you see, let's just double check that everything's going okay. Yeah. And then, okay, then we ran this. So on the one hand, we have to pick up the KOs that are interesting. So yeah, actually this makes a lot more sense. Yeah, so we pick up the KOs that are interesting, and and then pick up the uni genes that have a hit to that, and we need to go back to the sequence just to get their length, because the length is what we're going to be using here. Then we put those things together and we do what we did in the past. And so actually here I am going to do a little bit of magic, which means I'm going to pause the recording and then pick up in a few seconds. Okay, and that was a bit of magic because I just paused the recording while this was running, because this takes a long time. And actually that was something that we decided that we would work on the next session. So then if we do this, so now we have the GMGC as our basis for evaluating what z-scores are. So let me just see here. This is the same code as before. So we've also decided that, or I decided that, just because of how this has grown a little bit without just random code addition, so it really does need to be cleaned up and refactored. So for example, all of this code that I had to pause the video for, which runs parsing the big FASTA file, this should have been done one time and then saved. So we will do a lot of that, just clean up next time. But in the meanwhile, this is what we had. Let me just add here also, I need to add a little bit of that. And then if we do this, z-scores, so this is exactly what we had done before, except now the z-scores are using the GMGC as the reference. And so now, somewhat surprisingly, there's these very, very low g-scores in the wild type already, but at the same time, if we do this at the same time, if we look at the at the same time, we also have the difference between what we see in the wild type and with 1,000 mutations. It's actually very large. And if we run this, I'm just going to run this over all of them. So we did this. Then we see, first of all, see the plots. So, oops, okay, oh, that's the, yeah, we see, again, that across the different, so this is Psilus subtilis, we see a lot of, a big enrichment in the 1K, but even in the wild type we see these somewhat surprising more than when we had used the uniref. So I think we're making progress. We also did start to explore just what some of these outlier KOs are. So some of them did seem to be ones that might not be so usable, but other ones. So we kept making progress. We would ideally like to find that if some KOs have a lot of variance, maybe we don't use them. But we are making progress. So here, this was a little bit surprising, but we can see big shift compared to 1000K to the wild type, so 10-fold increase. So that's actually, so this was E. coli. All right, and so next time, what are we going to do? So we are going to be first of all just starting to clean up things. This grew very organically, very exploratory, but it's now, and it takes a while to run. It's generating a bunch of plots that I'm not even looking at so much anymore. So let's try to clean this up a little bit. Let's start to get some of these computations that took 15 minutes. I even paused the recording. Maybe we put them in a file that runs once as part of the jug process, and then that's done. And maybe some of these things, instead of being printed to the consoles where I have to rerun them, we can run them one time and save the results. So but you can see it's pretty good. And if we can now find some of the KOs we can reject a priori as not being good, these outlier KOs that are we could potentially get really good results. Right, so with that, I'm gonna remind everyone that everything is always on GitHub. We also have this mailing list, we have Discord. So next week I'm not going to be here, but I decided that instead I'm going to pre-record instead of stream, and then I'll just schedule it for the normal streaming time. So that will be a little bit of a different way of approaching things, but hopefully still keeping with the spirit of extremely open science. So everything will be open, everything will be streamed, everything will be public. So you maybe you won't be able to join the stream live, but you'll be able to give us commentary on either the mailing list or on Discord. All right. So with that, have a good weekend, everybody. See you next week.